Good morning. Today's Grand Rounds is very special in multiple ways. First, it is a memorial lectureship to one of volunteer faculties who did a lot for the program for many years. And the second is the speaker. Is a very unique speaker that I'm grateful that he agreed to come to us. So as always, a word about the honoree Dr. Don Fournier, and one of the previous fellows, actually was the first fellow when I came here, Dr. Kurtz Hedberg, who's practicing in Arkansas, has been very successful, and he was instrumental in initiating this lectureship, and he always comes to attend it, so I'm grateful to you, Kurt. Kurt has been wonderful. I'm proud of him for his success in his practice. He added 3 partners already. He's active nationally, particularly on the Board of Regents of the American College of Allergy, and I'm expecting for him more national recognition. Kurt, please may you come to introduce Dr. Fournier.
Thank you, Sam. So, Don Fournier, a great teacher, a great lecturer. We're here to honor him today. Passed away in 2008. A 1974 graduate of the University of Texas Medical Branch in Galveston. He did internal medicine residency in allergy immunology at Welford Hall in San Antonio. He's certified by the American Board of Internal Medicine and the Board of Allergy and Immunology. He was busy in a number of different societies, the Louisiana State Society, the Texas Allergy Society, as well as the American College, the American Academy of Allergy, and the American College of Physicians. He's a member of the editorial board for the Allergy and Asthma Proceedings, and was a retired lieutenant colonel in the U.S. Air Force. In addition to his solo private practice, he actively participated in many scholarly activities, and that's what we're here to memorialize today. For many years, he served at the program here, and then also in committees at the Southwest Allergy Forum and the Eastern Allergy Conferences. He was a past president of the Texas Allergy, Asthma, and Immunology Society, and governor of the regional, state, and local allergy societies in our region. He was volunteer faculty here at LSU. He contributed significantly to the allergy and immunology program here, with training a number of different allergy fellows for over 20 years with great dedication. He would come most every Friday and sometimes even other days during the week. He passed away after a short illness on February 9th, 2008 at the age of 58. And to commemorate his contributions to this department, we've established this annual memorial lectureship, and I appreciate you all being here today.
Thank you very much, Kurt. Well, I am honored to introduce the speaker for today. We always get guest speakers from all over the nation, very prominent people. Today's speaker has extra because of not just his scientific stature nationally and internationally, but because of his special friendship with me that I treasure. Dr. Robert Lemanske has got his MD from Wisconsin and got the pediatric residency also at the University of Wisconsin, allergy immunology also at the University of Wisconsin, and continued working at the University of Wisconsin after further got an additional fellowship in allergy immunology at the National Institutes of Allergy and Infectious Diseases. He's board certified pediatrics and allergy immunology, has extensive basic and clinical research, particularly on the pathophysiology and the treatment of asthma during early childhood. He is currently a professor of pediatrics and internal medicine in the Division of Pediatric Allergy and Immunology and Rheumatology at the University of Wisconsin and Medical School and Public Health. His associate recently has been selected to be the Associate Dean for the Clinical and Translational Research, a witness to his great contribution and research at the institution there. He was a former director also of the Morris Institute for Respiratory Research at the University of Wisconsin for 9 years. He's the immediate past president of the American Academy of Allergy, Asthma, and Immunology, which only a very few people can reach that status. He completed his presidency earlier this year, and in this picture passing the gavel to Dr. Tom Fleischer, who a few years ago was also a speaker here. He is from the National Institutes of Health. He published more than 200 articles in the most prestigious journals and contributed more than 50 book chapters and co-edited 3 textbooks. He's on the editorial board of JACI and Pediatric Allergy Immunology and possibly others. He was served on the board of directors of the American Board of Allergy Immunology, and he also became the chair of the board. He served on many major national committees, NIH interest sections, and the National Heart, Lung, and Blood Institute Advisory Council. He's very instrumental in developing and updating the national asthma guidelines, not just in the United States, but collaborated with the European societies as well. He is a sought-after speaker at major conferences on allergy, asthma, and respiratory diseases. And it was not easy to make him come here. So I just told him, you will come, just to choose whatever date, because every date I gave him say, I am not available, I'm traveling here and there. He said, you are going to come. He has many national, international honors and prestigious awards. Most recently, just a couple of weeks ago, he got the prestigious Brett Ratner Pediatric Allergy and Immunology Award by the Academy of Pediatrics section on allergy and immunology. Only a few people deserved that. One of his books that he co-edited is the, in capital The, Bible of the book on allergy and immunology, large 2 volumes. He is among a very selected group of colleagues in that important book. With all these achievements, he has his fun side. We always think that the person of that stature will be just bookworm and dull. It's not Rob. So he's just there. He didn't ask me to join him here, probably that I don't fit with that group, but it's just great. I have many good memories with him over the years that I treasure, but I will share 1 or 2 only. Where was that picture? Rob? This was about 25 years ago. Do you remember? If you remember, I'll give you $1. Where? Yeah, Deauville, correct. You got, I will give you the $1 when I get my paycheck at the end of the month. Deauville, France. It was a great trip to your a certain group there, and I enjoyed this very much. Everyone enjoyed it, and I was just in. That was Normandy area, and Deauville, and later to Paris. Good areas. So it made me. I liked it. So in a shopping area, I disappeared. And I found Dr. Lemanske coming running to look for me. Other people, I don't know whether they noticed my absence, but he definitely saw that I'm not in the bus. So he came to take me. And I really reluctantly followed him and joined because I was happy in that trip. The hotel that I was staying in there, I was in room 620, and the same floor, I find the Princess Stephanie of Monaco suite, and also next to me there is the suite of Elizabeth Taylor. And hey, I'm just writing in a piece of paper by hand something to stick it. The little room of Samibana, but they prevented me. So it was a good area. Why would I go back? But anyway, I yielded. That I can say in a way he saved me, although I wanted to be lost. And I say asylum to France. Another anecdote. We were, do you remember this?
Oh, yeah.
You do? Okay. We were in an international meeting on asthma in Japan, and he kindly said he was invited by a friend to a special Japanese dinner. And that day was the banquet dinner for the whole conference. I told them we leave here and we go for that special. He said, yes, because it's a very special dinner, it's a very special restaurant, not the communal mass food here. And I said, yeah, but the guy that you're going with him doesn't know me. He said, no, but he said, if you have friends to come. And I don't know whether I was the most loved friend of his or I was the only friend that he took me. Anyway, the guy drove us there, took us in a dim restaurant. We sat on the floor, the table is very low, sat with great difficulty. And the first course came, the bowls came. And his late wife was between me and him. And I found in my bowl something moving. So I told her, did you see that? She said, oh my God, something moving. So we did not eat except bread, but I think you ate everything. We ate bread, we could not eat. But I forgave him for this. His intention was good, and he saved my life in France. I said, all right. But it's wonderful to have this anecdote with you. We have been in many, many places that I treasure, Rob. With this, it is really a great pleasure and appreciation that you came to give this lecture, Rob, and please, you will come to switch. And on this occasion, there is a commemorative plaque for you.
All right. Well, thank you, Sammy. Those are some really, really wonderful memories. I value your friendship a lot, and I'm very, very grateful that you asked me to come. And it's a great honor to be able to give this memorial lecture. So with that, what I'm going to try to do in the time that I have is to address this particular question, and that is, when and how does asthma begin? And to do so, I'm going to review with you a number of different areas that I think are very, very important to keep in mind. We know that there seems to be some critical developmental windows in preschool children that eventually leads to the expression of the asthmatic phenotype. And there are genetic components that are important. I'll review with you some of the important genes, including a region in 17q21, another new chromosomal hit, if you will, the so-called CDHR3 locus. We're also going to go over some of the environmental exposures that I think are very important, some very new work with the Hutterites and the Amish population, and the importance of the microbiome in the expression of childhood asthma. Our group has been very interested in respiratory pathogens, including viruses, particularly the human rhinovirus. And I'll also review with you some of the newer data that looks at viral and bacterial interactions. As an allergist, I'm very, very keen on the fact that allergic sensitization, particularly probably in the first 2 to 3 years of life, is an extremely important event that we feel is very important, along with the development of wheezing, lower respiratory illnesses, that somehow this combination in the context of a genetic background and also other environmental exposures that ultimately lead to the expression of childhood asthma. Now I'm going to divide my talk into the following 5 areas. First, I'm going to briefly review with you the so-called concept of wheezing phenotypes in early life, go over with you some of the work that's been done, particularly in the United Kingdom, looking at the airway inflammatory events that occur within the first 2 to 3 years. I'll address alterations in lung function, spend a little bit more time on environmental factors, particularly infections and the development of allergic sensitization, and finally talk about genetic contributions, and if time, we'll talk about how all these things are important to be thinking about when we approach treatment in this age group. But more importantly, is it possible, knowing all these things, if we could actually prevent the development of childhood asthma? Well, let's look first at the clinical expression of various wheezing phenotypes. And there seems to be a demarcation between what happens in the first 3 years and what happens after age 3. This was some work that was originally proposed by our Tucson colleagues, looking at the expression of various wheezing phenotypes. And they had reported in 1995 the so-called transient early wheezer. These are kids that wheeze in the first 2 to 3 years of life, but then stop. Then there's a group of kids who are late onset wheezers. They don't wheeze in the first 3 years of life, but then begin to do so and continue in later childhood. There's a group of kids who are the persistent wheezers. These are kids who wheeze in the first 2 to 3 years and just keep going. And for all practical purposes, these are the kids that go on to get childhood asthma. But using a very large data set from our colleagues in Scandinavia, there were an additional 2 wheezing phenotypes that were discovered, the so-called intermediate type, which unlike the transient wheezers or the late onset wheezers, which basically go away or get better, these kids continue to be symptomatic. And then another group, the prolonged early, these kids wheeze early on, but they don't really stop at age 3, but eventually they do go on to go into what we might call remission. What about the histologic expression of childhood asthma? And I'd like to review with you some of the findings that have been noted. First, in infants, that is in the first year of life, the eosinophilic inflammation and reticular basement membrane thickening that we really associate with the histopathologic changes in adult asthma are not yet present, despite the fact that various investigators have been able to demonstrate that if you do pulmonary function testing in early life, you can actually demonstrate reversible airflow obstruction, but these inflammatory changes are not yet present. So this strongly suggests that the physiologic expression of asthma actually begins before the inflammatory events. However, by age 3, both of these abnormalities are present, particularly in children who have more severe persistent wheezing. 2 other features, increased angiogenesis and epithelial shedding, have also been reported in the first 3 to 4 years of life. And importantly, these markers of airway wall remodeling and inflammation don't differ between episodic versus multiple trigger wheezers, meaning those kids who only wheeze when they get a viral infection versus those kids who wheeze with allergen exposure or exercise, etc. And then a new finding, which I think is important, and that is airway smooth muscle. And this appears to be increased only in the kids who wheeze and have allergic sensitization. And importantly, this increased airway smooth muscle mass is the only reported feature to be a predictor of the development of future asthma. Well, what about the physiologic expression? And what I'd like to review with you very briefly is alterations in lung function. And again, this is some work that was reported by our Tucson colleagues in their birth cohort, looking again at the kids who never wheezed. This is looking at z-scores of lung function, age and years along the x-axis. And what you see here is the transient wheezers actually look or have diminished lung function in early life. And that's probably why they're wheezing, because their airways are smaller in diameter. So when they get inflamed or there's increased mucus, they begin to make noise. Eventually, however, they go on to recover. And the group that's most concerning are the persistent wheezers. These are kids who are born with nearly normal lung function, but then in the first 4 to 6 years of life, they lose significant lung function, which they appear never to regain. Now, this is some work that we have generated in our COAST study, which is an acronym which stands for Childhood Origins of Asthma, trying to determine what factors may be important in causing this loss of lung function in the early part of life. And this is looking at kids. This is FEV1, or forced expiratory volume in 1 second. This is age in years. These are kids who wheezed in the first 3 years of life related to human rhinovirus, and these are kids who wheezed with RSV. And very interesting, those kids who didn't wheeze, as opposed to those kids who wheezed with rhinovirus, the kids who wheezed were those kids who had significant decreases in lung function in the first 5 to 8 years of life. In contrast, those kids who wheezed only with RSV, really there was no significant difference in lung function, whether they wheezed with RSV or did not. Now, in addition to the viral pathogen that's causing the problems, we also looked at the frequency and the severity of these exacerbations. And what we found was something very interesting. The green dots are those kids who never wheezed with rhinovirus, those kids who had mild to moderate wheezing in yellow, those kids who had one severe exacerbation, and we're defining as severe exacerbation, are the need for oral prednisone, and the red are those kids who had 2 or more severe exacerbations. And both with FEV 0.5 and FEV1 at the various years that we did the pulmonary function assessment on our children, you can see that 2 or more of these severe exacerbations were associated with the loss of lung function over time. So the importance of these observations is, number one, the type of insult, that is, the viral pathogen, appears to be important, and also the frequency and the severity of these so-called hits, if you will, in the lower airway tend to cause problems or be associated with problems over time. So how is this happening? Well, this is some work from our Canadian colleagues who looked at chemokine release from human rhinovirus-infected airway epithelial cells and being able to demonstrate that this interaction actually promotes fibroblast migration. And as you know, fibroblasts in the airway have a great deal to do with airway wall remodeling, the stiffness of the airways, which we associate with more severe episodes or severe asthma, particularly in later childhood and in adults. And the 2 big drivers of this appear to be a chemokine called IP10 and also a chemokine called IL-8. And interesting, interleukin-8 is a very potent neutrophil chemotactic factor. And what this group concluded from their work is the data support the concept that recurrent human rhinovirus infections can contribute to aspects of airway remodeling in asthmatic patients. So what about environmental influences? And what I'd like to focus on are the influence of respiratory illnesses. Now, as pediatricians, we all know that there are kind of 2 groups that we see in our clinics. There are healthy infants or young children. They can get viral respiratory infections. They then develop upper or lower respiratory symptoms, but they go on to resolve the problem. In contrast, in the so-called predisposed infant or young child, they may have underlying allergic inflammation, which I just went over with you. They may have an impaired epithelial barrier. They may, and we have evidence, and other groups have evidence to support, that there may be an impaired innate immune response in terms of a diminution in the release of interferons, which are very important in protecting the host from viral spread. And in this context, when these kids get the virus infections, they do develop lower respiratory illnesses, but now something different happens. This can lead to airway damage, airway wall remodeling, which I've already demonstrated to you, and repeated infections can cause recurrent wheezing and ultimately lead to the expression of the asthmatic phenotype. Now, this is some work that my colleague Dan Jackson and our group published in the Blue Journal in 2008, looking at RV wheezing versus RSV wheezing in the first 3 years of life, and the chances of these kids going on to be diagnosed with childhood asthma by the time they were 6. Now, as we all know, RSV can be associated with this particular outcome, and indeed, we found it was a risk. But at the time we were doing these analyses, we were able to really detect human rhinovirus, which was not possible previously. And what we found was something very intriguing, that the kids who wheezed with rhinovirus were very significantly increased in terms of their risk of going on to getting asthma. And if they wheezed with both, that risk was not substantially higher. Well, we also became interested in some of the work that has been published by other groups. This is again by the Tucson group. Now, when they were looking at these illnesses, they were not able to really detect human rhinovirus based on the molecular technology that was available at the time. And what they did is they looked at those kids who wheezed with RSV in the first 3 years, and if they had infrequent wheezing as they got older, or if they had frequent wheezing as they got older. And what they noted is, at least for the first 10 years of life, these antecedent RSV wheezing illnesses were associated with the persistence of the wheezing, at least out to age 10. But by the time these kids got to be adolescents, this effect pretty much dissipated. So we took our COAST cohort to look to see if we could see similar results. And on top of that, if human rhinovirus would be associated with more persistent effects. And we did an analysis looking at the odds ratio for asthma at age 6, 8, 11, and 13. And the red are those kids who wheezed with rhinovirus at some point in the first 3 years. And the green are those kids who wheezed with RSV. And once we adjusted for wheezing with all viruses, what we found is very similar to what the Tucson group. By the time these kids got to be age 13, we no longer saw a significant effect with the antecedent RSV wheezing. In contrast, if the kids had wheezed with human rhinovirus, the effect on long-term asthma outcomes appeared to persist to a much greater extent than we were seeing with RSV. So this indicates that there's something really different or important for us to be thinking about in terms of rhinovirus illnesses in early life. And this is a human rhinovirus tree that was published, a phylogenetic tree that was published by our colleague Ann Palmenberg at the University of Wisconsin, looking at genetic similarities and eventually genetic differences in terms of where the various types of RVA graph out, where the various types of RVB. And we knew for many years about human rhinovirus A species and B species because they bound to either ICAM1 or low-density lipoprotein receptors. But then eventually laboratories got more smart, if you will, and they began to grow another species of rhinovirus. And in the last decade, we have learned that this particular species may be very important in terms of asthma pathogenesis. Now, the reason it took so long to find C was because it wasn't binding to the various receptors that we knew that A and B bound to. And what I'm going to show you in the next few slides is how we came about to learn what the particular receptor is for human rhinovirus C. Now, from a clinical perspective, why is HRVC important? Well, this is looking at some work from our Australian colleagues, looking at a bunch of children who were brought into the emergency department for acute lower respiratory illnesses. They were cultured at the time they came into the emergency department and then followed for a number of subsequent months to see what these early events did in terms of recurrence of admission or recurrence back for an acute episode of lower respiratory illnesses. And looking at all viruses, this is the regression curve. And looking at human rhinovirus C, you can see there's a huge difference in terms of those kids who were infected with HRVC and their recurrence risk in terms of coming back into an emergency room setting for an acute lower respiratory tract event. Now, importantly, in their data set, 53% of these children were atopic or had demonstrable allergic sensitization. But even more important, in atopic children, HRVC was associated with a 6-fold increased risk of subsequent admission. And I bring this up to you now because in a couple of slides, we're going to go over the importance of allergic sensitization. And I want you to keep in mind that this combination of HRVC and allergic sensitization in terms of risk of morbidity in childhood asthma. So as I mentioned to you, we knew that human rhinovirus A and B bound to ICAM or low-density lipoprotein receptors. And then Yuri Bachkov in our laboratories, along with Jim Gern's lab, my colleague, began to investigate what the receptor was for human rhinovirus C. He determined that it was a unique receptor. It was not expressed on undifferentiated epithelial cells and most cell lines, but it was indeed expressed on fully differentiated epithelial cells. So then along came this particular article from our colleagues in Denmark who did a genome-wide association study and identified CDHR3 as a susceptibility locus for early childhood asthma in those children who went on to get severe exacerbations. So Yuri began to think about a possible connection between what CDHR3 was coding for and what HRVC may or may not be binding to in the airway. And lo and behold, what he reported in the Proceeds of the National Academy of Science in 2015 is that cadherin-related family member 3, which is a childhood asthma susceptibility gene product, mediates rhinovirus C binding and replication. Now, what is CDHR3? It's a transmembrane protein with as yet unknown function. It enables RVC binding and replication. And a coding SNP in CDHR3 I just showed you on the previous slide, previously linked to wheezing illnesses and hospitalization, also mediates enhanced RVC binding and progeny yields in vitro. So this is an important finding in that as we talk about asthma prevention, if drugs that don't block this particular receptor, but do block receptors for A and B, clearly we're missing out on about 1/3 of the rhinovirus population. And if we're thinking about asthma prevention and the importance of rhinovirus wheezing illnesses, this may be a receptor or a target that we need to be focusing more attention on. Well, what about immunologic expression? And there's lots to talk about here, but for the lack of time, I'm going to focus on the development of allergic sensitization and answer first this question: what influence does allergic sensitization have on asthma risk? This is again some work that Dan Jackson published from our coast cohort, looking at RV wheezing and allergic sensitization in year 3 and asthma risk at age 6 years of age. So what we're graphing here are kids who wheezed at any time with rhinovirus in the first 3 years, and then a cross-sectional look at whether the kids had one or more positive in vitro IgE test to aeroallergens or not. That's what RAS positive and RAS negative means. So the control group, those kids who never wheezed with rhinovirus and who were RAS negative, their odds ratio is obviously going to be 1. If they had demonstrable allergic sensitization, but we could not demonstrate that they had wheezed with rhinovirus, their risk was increased to about 3.5. So clearly just the presence of allergic sensitization seems to increase risk. But then look what happens if they're RAS negative and they have wheezing with rhinovirus. This risk substantially increased. And if they have the double hit, if you will, they have histories of wheezing with rhinovirus and they have allergic sensitization develop by the time they're 3 years of age, their risk of going on to getting asthma is significantly increased. So the next question we asked is, what influence does age and the magnitude of sensitization have on future asthma risk? And this work was originally reported again by our Australian colleagues in their cohort, basically demonstrating that if the kids were sensitized in the first 2 years of life, very important, first 2 years of life, they were at significant risk of going on to getting frequent, recurrent wheezing and also the diagnosis of childhood asthma. So this is some work by one of our fellows, F.J. Rubner, which is currently in press in the JACI, looking at our COAST cohort in terms of early life rhinovirus wheezing, allergic sensitization, and asthma risk by the time these kids were 13 years of age. And this is small print, but the orange are those kids who were sensitized very early on, by the time they were 1 year of age. The yellow are those kids who were unsensitized, not sensitized by age 1, but were sensitized by the time they were 5 years of age. And then the kids who were unsensitized by age 5 years. And I think you can see just the age of sensitization here is extremely important in terms of conferring increased asthma risk. In addition to the time this happens, the quantity by which it happens also appears to be important. This is looking at the number of sensitizations, the age at which these kids were evaluated. So again, as these kids get more and more aeroallergens to become allergic to, their risk continues to increase. And so we concluded from this that both the age and the number of sensitizations influence the subsequent risk of going on to getting asthma. Well, we also wanted to know which comes first. Does allergic sensitization come first, or does viral wheezing come first? And what our biostatisticians put together is this two-stage model in which neither state was achieved. The kids began to wheeze first with viral respiratory infections, or they became sensitized first, and eventually both had occurred. And the way this model works, if viral wheezing caused or preceded sensitization, we would predict that movement from stage 2 to stage 4 would be greater than movement from stage 1 to stage 3. In contrast, if sensitization causes or precedes viral wheeze, we would anticipate from the model that movement from 3 to 4 would be greater than the movement from 1 to 2. And what we found, and we published again in the Blue Journal, is that sensitization leads to viral wheezing or precedes it, and the reverse does not appear to be true. And this is looking at all viral respiratory pathogens in terms of the risk. This is specifically looking at human rhinovirus, and this is specifically looking at RSV. We also now have looked at other type 2 inflammatory markers in early life in terms of whether they could inform us regarding future asthma risk. And this is some work that one of our fellows, Haley Anderson, has put together, and this is also in press in JACI, looking at the relationships among peripheral eosinophil counts, aeroallergen sensitization, and serum periostin on the development of childhood asthma. And this is looking at the odds ratio for asthma at age 6, and this is looking at age 11, and these are the values for eosinophil counts greater than or equal to 300. You can see that's risk increases risk, and this was all evaluated at 2 years of age, and it continues to be a risk out to age 11. Any sensitization in terms of allergic sensitization also increased risk at age 6 and still at age 11, and increased periostin levels, which have been felt to be a type 2 inflammatory marker. When we looked at this for asthma risk at age 6, it was associated with that, but it was no longer a risk by the time these kids got to be age 11. So the importance of this clinically is that if you don't have access to allergy testing for whatever reason, getting an eosinophil count in early life can potentially help you decide what the relative risk of these wheezing kids could be in terms of them ultimately expressing an asthmatic phenotype. Well, what about interactions between viruses and bacteria? First, looking at asthma inception, this was some work that was published again by our Danish colleagues in which they did a nasopharyngeal swab at a month of age and looked for various bacterial pathogens and then followed these kids out to age 5. And what they found is if these kids were colonized with strep pneumonia, M. catarrhalis, or H. influenza, they were at greater risk of developing persistent wheezing out to age 5 compared to those kids who were not colonized. Then this is some work from our colleagues in Australia looking at a birth cohort that's very similar to COAST, looking at year 1 nasal secretions at 2, 6, and 12 months of age in 487 healthy specimens and 534 sick specimens. And they did a microbiome analysis with the 16S gene deep sequencing, looked at viral pathogens, and characterized the illness as either being febrile or not. Now, this is a busy slide, but it looks at the nasopharyngeal microbiome in infants and the risk of respiratory illnesses. And what I want you to just see here, look at this top part of the graph. During acute respiratory illnesses, these kids were much more likely to have H. flu, strep, or varicella compared to those kids when they were well. In contrast, when they were sick, these guys, I can't see this from here, Aloecoccus, Cornybacterium, or Staphylococcus, these kids had much less of these particular bacteria compared to the kids when they were well. So this suggests that if we could somehow get rid of these guys and get more of these guys in the airway, we could potentially tilt the scales, if you will, into less severe illnesses, respiratory illnesses in this early part of life. Now, they went on to follow these kids out to age 5 to 7, and they found that the risk factors for future asthma were first, if they had early colonization with strep, second, if they were febrile during the illness, and here we go again with HRVC, if they could also detect this particular species of human rhinovirus. What about the microbiome and innate and adaptive immunity? Well, this is some work that I hope that you have seen, and if not, I'll briefly review it with you. This is looking at innate immunity and asthma risks in Amish versus the Hutterite farm children, and by a group of investigators that are great colleagues of ours in allergy and immunology, including Fernando Martinez, Donato Vercelli, Carol Ober, who does all of our genetics for us for COAST, and Erika von Mutius from Germany, who has done a lot of work with the microbiome in farming communities. So to set the stage, both the Hutterites and the Amish, and I'll show you this in the next slide, really share genetic heritage. They're very similar genetically. But when they moved to the United States or emigrated to the United States, they developed 2 very different styles of farming. The Hutterites have developed a much more modern way of dealing with their farming environment, as opposed to the Amish, which are much more primitive in terms of their barn structures, how close their houses are to the barns, et cetera, and also when the kids in early life are allowed to go into the barns as opposed to the Hutterites, where the kids really, the barns are so separate from the homes, they really don't want to go into the barns as opposed to the Amish children. So this is the demography of what they studied. Importantly here, this group of investigators, the Amish, they could not find a single child who had asthma, which really surprised them. So they felt, well, let's see if we can figure out why these differences exist. So they studied 30 Amish children, 30 Hutterites, 0 asthma in this population. 6 out of the 30 Hutterite children had asthma. Now, as I told you before, this in blue is the analysis of their genetic background. The blue is the Amish, the red is the Hutterites, almost right on top of each other, as opposed to these other different ethnic backgrounds, the Tuscans, the Russians, North Italians, etc. And the purpose of this slide is to basically emphasize that the genetic background of both the Amish and the Hutterites are extremely similar. In contrast, when this group looked at the genetic expression profile in peripheral leukocytes, they were very different. So the red here, these are genes that have higher expression in the Hutterites relative to the Amish. The blue are those who have a higher expression in the Amish relative to the Hutterites. The black, there is no difference between the populations. So despite having the same genes, the ability to turn on different sets of genes is very different in these two populations, suggesting that there's something in the environment that's causing this differential expression. And indeed, when they looked at the environments in both the Amish and the Hutterite children, when they looked at endotoxin levels in airborne dust, the Amish had a much greater level of endotoxin in their dust compared to the Hutterite homes. Now, they also began to drill down to determine what possible, why these expression profiles were different and what biologic significance that this would have. And this is looking at genes that were more highly expressed in the Amish children. And I've circled here some of the important pathways that they wanted to emphasize. First, this TNFA1P3. It encodes for A20, which is an enzyme that inhibits multiple inflammatory pathways that are dependent on NF-kappa B signaling. NFA1P3 also has been shown to mediate the protective effects of European farm dust extracts in mouse models of allergic asthma. And the IRF7 particular gene regulates type 1 interferon transcription and is therefore essential for innate airway responses against viruses that are linked to susceptibility to asthma in early childhood. And what they did then is they took their experiments into a mouse model in which mice are sensitized to ovalbumin. They're then challenged with ovalbumin. When they're challenged with ovalbumin in the airway, they develop airway hyperresponsiveness, which you can measure by giving them aerosols of acetylcholine and measuring airway resistance. So the greater the airway resistance following exposure to acetylcholine, the more airway reactivity is present. And what they did is, these are the mice here in gray that were sensitized to phosphate buffered saline, or PBS. That's the control group. Then there's the group that was sensitized to ovalbumin, and you can see the sensitization and the challenge with ovalbumin produces increased airway reactivity, which we see in asthma. They then had these mice inhale dust from the homes of the Amish families, and look what happened. Their airway reactivity significantly decreased. In contrast, if they had the mice inhale dust from the Hutterite homes, their airway reactivity actually increased. They then went on to move their model into a different strain of mice because they could do knockout experiments with this particular strain. And what they found is if they knocked out two genes that are very important for innate immune responses, if they knocked out one of these genes, the protective effect of this Amish dust tended to go away. And if they knocked out both of these genes, it totally went away. And so what they concluded from this is that innate immunity has evolved to sense the environment and transduce signals that calibrate adaptive responses to exogenous antigens. And this MYD88 and TRIF locus are located at the convergence of multiple innate immune signaling pathways, and deletion of these molecules virtually disabled innate immune responses in these animals, and thereby dysregulated the development of adaptive immunity. And I don't have time to show you this, but a lot of the findings that they had generated in both the Amish and the Hutterite kids in terms of innate immune responses were exactly what you would have predicted based on these animal experiments. And they therefore concluded that susceptibility to asthma may be enhanced when innate immune system responses to environmental stimuli are weak or dysregulated. And finally, just ending up with a gene experiment that we did with the Coast kids. I mentioned to you at the beginning of my lecture that the 17q21 region seems to be very important. And within this region is a gene called ORMDL3. And we're interested in looking at what this gene does and why it may be important in the expression of asthma. And the reason we are is that the 17q21 locus is the most replicated asthma locus and represents the most significant genetic risk factor for childhood asthma known to date. And what we found and we published in the New England Journal of Medicine is that if we look at this is one particular SNP in the 17q21 region, looking at the various allelic expressions at this particular locus, CC, CT, and TT. When we looked at asthma prevalence from years 6 to 8 in our cohort, if these kids did not wheeze with rhinovirus in the first 3 years of life, regardless of what genotype they had at this particular locus, their risk of going on to getting asthma was exactly the same. Interestingly, however, if they had one T allele at this particular locus, their risk of going on to getting asthma, if they wheezed with rhinovirus in the first 3 years of life, was substantially increased. And if they were homozygous TT, their risk of that going on to getting asthma was close to 90%. And there was a significant interaction between the genes that these kids had and the pathogen by which they were infected with and led to wheezing illnesses. So in conclusion, I've given you something I hope to think about in terms of when and how does asthma get started. We just went over the 17q21 locus. I reviewed for you the CDHR3. We looked at the microbiome in the Amish and the Hutterites. We went over the importance of various respiratory pathogens, particularly HRV, and more specifically HRVC, some very new developments. The importance of virus and bacterial interactions in early life, the very, very important development of allergic sensitization and how this interacts with wheezing, respiratory illnesses, and eventually the expression of childhood asthma. So with that in mind, if we want to prevent the development of childhood asthma in early life, what could be some of the mechanisms or ways we could do this? Well, first, we could target IgE, and with using omalizumab, which is a monoclonal antibody against IgE. And we and other groups have now been funded by the National Institutes of Health, and we will be starting in January of giving kids down to age 2 this monoclonal antibody to see if we can actually prevent the development of childhood asthma. I showed you some data to suggest that the human eosinophil in the peripheral blood is a marker for the persistence or the expression of childhood asthma. And what about targeting IL-5, which is a very important cytokine in terms of the ingress of eosinophils and prolonging the life of the eosinophil in various target tissues? Could we target the genotype? If we knew what these kids were at the 17q21 locus and we wanted to design prevention trials, obviously if we knew what the risk alleles were, parents would be much more likely to enroll their children in prevention studies in terms of trying to diminish this potential risk of developing asthma. We could also think about preventing infection with respiratory pathogens. There's already a vaccine out for RSV, and various groups are now looking at vaccines for rhinovirus and hopefully being able to develop drugs or methods of being able to block this human rhinovirus C receptor. I showed you data from the Amish and the Hutterites and thinking of ways that we may be able to enhance innate immune responses, and finally altering the microbiome. Can we target pathogenic bacteria from some of the work I showed you from Australia? And we and other groups have now been funded by the National Heart, Lung, and Blood Institute, and we are beginning a study in December called the Orbex study, in which we will be giving lyophilized bacterial extracts to children in the first 6 to 18 months of life who had increased risk of going on to getting asthma, to see if we can change their microbiome in their guts. And the gut talks to the lung in terms of influencing the microbiome in both of these different target organs. And that work will be going on for the next 4 to 5 years. So in conclusion, the other things that we need to be thinking about, if we do find the right particular method or right particular intervention, who is the right patient for us to be using these interventions with? Hopefully we'll know what the correct treatment is and what our options might be. When is the right time to treat? Like in the first year, in the first 5 years? How long do we need to treat, and what do we need to do to monitor? And finally, if we're using very expensive interventions like monoclonal antibody, how do we also put into the mix the cost of the therapy and the potential side effects over time? So with that, I'd again like to thank the group for inviting me. It's a great honor for me to give this lecture. And I want to also acknowledge Sammy's friendship, and I hope it'll continue for many years to come. Thank you.
Only you can give this lecture, Rob. Any one or two short? Of course. Aaron.
Thank you. Thank you for trying to address this complex subject which has been around for a long time. What fascinated me was your slide from Tucson, and I was faculty there before I came here. When Taussig started that longitudinal studies, to my knowledge, he was looking at premature baby, and then the other folks took over when he moved to Denver, Colorado, heading the Children's Asthma Research Institute. So the issue, what fascinated us is why are certain subgroups of premature baby that go on to develop asthma, as you showed in your slide, versus others who have just wheezing. In other words, this subgroup would respond to methacholine challenge test when you test them later on in life, 5 to 7 years of age. As you know, he's a pulmonologist. So they've done fascinating work there. So that's number one question, that where do all these complex interactions between nature and nurture come into the premature babies, since I'm a neonatologist. Number two, has anybody looked at the pure population in North, for instance, in Greenberg, where they are a very pure population, they don't intermix, there's no intermarriage of gene polymorphism and existence of asthma, because they have certainly a high incidence of viral infections up in North.
Yeah, there's actually a lot of different cohort studies, as you point out at the beginning. These are very, very complex issues and plenty of interactions occurring between the host and the environment. I don't remember the Tucson study they may have started with premature babies, but eventually the cohort that has been reported in most of these studies is a group of kids who were not at high risk. These would be what would be called normal risk. And that differs from the coast kids and some of the other data that I showed you in terms of these kids are high risk because they have a mother or father who has asthma, allergies, or both. And the reason we studied high-risk populations were because of the amount of money we were getting from the NIH. We could only study so many babies. But when we looked back at our findings in coast and compared them to what the Tucson group had reported, we were pretty surprised that our findings were pretty close to what they had reported in a so-called normal population. Now, in terms of the genetic studies, I think this particular paper that I went over with you, it's very fascinating because the genetic background of these two populations is identical. And you saw that the dots are superimposed. But yet when you look at gene profile expression, they're very different. So there's, despite the fact that they have the same genes, there's something causing these genes to be activated in different ways. And you can imagine, I just emailed Carol yesterday, and they're hot after trying to figure out what is in this dust and in these mouse models that is preventing these things from happening. And it's very different in the Amish versus the Hutterites. And for those of you who read the New England Journal of Medicine, in this, I think, week or last week, there were some letters to the editor asking if they could expand on some of their findings. And one thing they didn't report on in the paper originally was, and I think I mentioned this to you when I got to that point, is that the Hutterite kids do not want to go in the barns in the first 1 to 2 years of life. They just don't. The Amish kids are living in the barns. So there's a huge difference there in terms of their exposures. And as an neonatologist and as pediatricians, my thinking on this is that this road is, or this pathway is to asthma is developed within the first 3 to 5 years of life. Now, not all asthma, we know there's different asthmas, but at least for childhood asthma, all of these things, we got to get there early, and we have to get the right targets, and we're not going to be successful in preventing it.
Well, can we benefit from the Amish experience to apply to prevent allergy?
Yeah, there's lots of work that's been done with farming communities. Erica von Mudius's work in Europe. There, the farming communities are very different than they are in the U.S. The barns are literally attached to the homes. So the kids are living, if you will, in, well, I won't say it, but they're living in all the stuff that the animals produce. And it's even a greater benefit if the mom is part of the process, of the farming process. If she's in the barn milking the cows while she's carrying the baby, the effect is even greater. So the Amish, I mean, I think it's the fact they have the same genes, they're both farming communities, but they farm very differently. And if the group can find out what this magic stuff is in the dust, I think that's going to be huge. We'll be giving this with nasal sprays.
So Kleenex against us.
Kleenex is against us, right?
All right, one last question. You have been called the ice cream doctor.
Well, when I was a kid, my pediatrician, he had an office above a drugstore that had a soda fountain. And so when I went to see him, he would give me a prescription for an ice cream cone. And so no matter how many shots I got or what happened to me when I was in the office, I always knew that I was going to get this prescription, and I could go down and I could get something that would make me happy. So when I started on the faculty at UW, we have some very famous ice cream there because we're America's Dairyland, and it's called Babcock ice cream, and they had it in the hospital cafeteria. So I had these prescriptions written up that say 1 ice cream cone or piece of fresh fruit, and then the SIG was to be eaten with a smile. And I would give these to the kids when they were done, and eventually they called me the ice cream doctor because of it. But we got to a point where not only did the patient get this, but all the brothers and sisters got one too. So sometimes the room was just full of kids, but it was fun, and I'm glad we did it.
Right, wonderful. Thank you.